أهلا وسهلا فيكم جميعا. أرحب بكم في حلقة الويبنار الخامسة في ويبنار المناعة في هذه الظروف الاستثنائية اللي نعيشها هنا في المملكة العربية السعودية وفي العالم ككل. طبعا شايف الأعداد ما شاء الله بتحضر، هذا شيء جميل. أحب أرحب بكم جميعا. أرحب بالطلبة، أخواني أخواتي الطلبة. أحب أرحب بزملائي الدكاترة. أحب أرحب بزملائنا من العلوم المختلفة. أحب أرحب أيضا بزميلتي اللي حتناقش أو حتكون معايا في طور هذا النقاش، زميلة عزيزة علي وخريجة مانشستر، أفضل جامعة في العالم، دكتورة آيات زواوي. كيف حالك دكتورة؟
أهلين مشرف، كيف حالك؟
الله يحييك، أهلا وسهلا. تعرفي بنفسك آيات عشان يعرفوك الناس؟
Sure. My name is Ayat Zawawi. As just Mushraf mentioned, I'm an assistant professor at King Abdul'Aziz University. So basically, as Mushraf just said, I'm his colleague since Manchester, actually. So yeah, I remember when you first told me about the webinar, I got very excited because I'm sure that we're going to meet an amazing people like you, Marcello. Thank you for accepting the invitation. I know you're working in the first line of defense, so I'm sure you learned a lot, and we're gonna learn a lot today from you. So I'm really looking forward for our discussion today.
Marcello, hello.
Hello.
How are you doing, my friend?
I'm good. How about you?
Not bad, not bad. How's the weather?
The weather is great. As I told you, it's gonna be Easter holidays for us,
Yes.
and the weather it's a mess. But since we are in a lockdown, the weather is great. So.
it's the irony of Corona, my friend.
Exactly. So.
Marcello, tell us a little bit about yourself first.
So yes. So first of all, I thank you both. I thank you the hosts of this webinar for this really kind invitation. And as I was telling before to one of the hosts, I think it's really important to have a knowledge sharing in science, in medicine, but it's important to share the knowledges across countries, especially across countries that don't have that much possibility to interact, like for example, a European country, Middle Eastern country, and I think that this is really important. So when Mushraf asked me to participate to this webinar, I immediately said yes, and really I thank you for this opportunity.
Absolutely.
So, first of all, I introduce myself. So I'm Marcello Albanesi, and I'm a medical doctor, and as for my formation, I'm a fundamental immunologist and a clinical allergist. Okay, so I think I'll need the board to tell you something more about me. So I come from Italy. I will share the screen with you, so you will see something. So basically, the first question that whenever you have to give a brief talk, or whenever you have to give a meeting, I think it's important to introduce yourself. So who I am. All right, so I'm an MD and I'm a PhD. Okay, so these are only letters that normally are attached to my names, but this has a particularly relevance when it comes to medical sciences. And this is because, so Mushraf told me that you all had immunology classes, and nearly all of you are working in the medical environment, either in biology or in dentistry or in medicine. So it is important to understand how the immune system works, because the immune system is basically a reflection of how our society works and how the things are done. So, and as a doctor, I think you all know that, for example, when you are in a tissue, okay, so this will be your tissue, and whenever you have a pathogen, that is bacteria, entering the tissue, this will be sensed by something that is called dendritic cells, okay? So, and you can interpret this as, if you want, in a society-wise, as the city, okay? So then the dendritic cells in an immune system function will migrate to a draining lymph node, that is the secondary lymphoid organs, and this dendritic cell will meet a T cell, okay? And this will generate a specific immune response. And this is an extremely particular feature of mammals, and only mammals have this, because plants have an immune system, but they can't do this process, and we can, all right? So, and basically you will generate either, for example, specific T cells or plasma cells that will produce antibodies, all right? So, and when it comes to medicine, you can say that everything that is here is medicine, okay? All right, you can consider the lymph node as the hospital, and normally, if you want to be a clinician, the clinicians are here, all right? So our clinicians are the effector arm of the immune response, okay? You go to the hospital from the city, and then you get cured, of course. But when I was a med student at the really beginning of my career, I realized immediately that this particular setting here that you see here was not enough for me. So I wanted to treat the patients, but I wanted also to be able to translate all the process that was happening in the city into the draining lymph nodes. So basically, everything that you see here is my job. So basically, MD, PhD, what it does, and in general, someone that has a formation in basic science gives you the possibility to translate the knowledges, okay? So you understand what's happening outside, you translate it, and then you can take precautions. Of course, many times the people are asking me why you are doing this. You can just be a doctor and earn a lot of money, but I don't think that this is the main point of medicine, okay? So we have, and you guys, all of you that you are listening, you are really lucky and having the possibility of studying how our body works, and this is a gift that you will have for all your life, and you have to use it in the more effective way that you can. So to give you, to tell you something about my background, okay? I've always been interested in immune system, okay? In how we can respond to pathogens or to external stimuli. And so at the beginning of my PhD, I will give you some details of what I did, and then we can dive together with Mushraf and Hayat into the specific questions about coronaviruses. But I think it's important for you to know what is my background in order to understand what I did and why I can talk about the immunology of coronaviruses today. So basically, during my PhD, okay, so this was, and by the way, this was the period of time where I met Mushraf during a summer school. And I was working on this project. So if you have a tissue, you all know that the tissue can develop tumors, okay? There is an uncontrolled cell growth. And sometimes tumors express on the surface some proteins that are tumor associated. So in medicine, I think you all are aware of this, but you can target this particular molecules with something that is called monoclonal antibodies, okay? So these monoclonal antibodies, what they do, I was working at the time with the breast cancer, and I was working with an antibody that is called in this way. It's called trastuzumab. Okay, trastuzumab, I'm sorry. That is NIGG1, and this is an important thing. So trastuzumab basically targets a molecule on the surface of the cancer cells that is called RBB2 that gives a positive kick in the cell. Okay, so basically this is a signal that tells the cells you have to replicate. And if you just put the antibody on top of that, this interaction will block this cell growth. But if you had immunology classes, and I think you all did, you know that an antibody that we can in an horribly way designed in this way, is basically shared into different portions. So you have the Fab portion that is what recognizes the antigen, and the FC portion that is basically what drives the immune machinery. Okay, so via the FC portion, what it does, it can activate the complement, and you can also activate immune cells. Okay, this is because immune cells have on their surface something that are called FC receptors. Okay, so if you have a tumor cell here, an antibody here, basically what you do is that you trigger the destruction of the tumor cells via the FC receptors. So, and to make a really long story short, because the PhD was 3.5 years for me, so my task at the time was to identify which cell subpopulation is responsible for the immune cell, for the cancer cell killing. And to make a long story short, after 3.5 years, I found out that the cells that are actually the killers are the neutrophils. Okay, and I will show you a video on that, and to make you understand the importance of it. And this is particularly relevant to all the kinds of infection that you have, because we all make antibody, and so the antibodies will function always in the same way. So in this video, you will see in red tumor cells and in green neutrophils. So if you don't do anything, as you can see here, the neutrophils will just pass by the tumor cells, and they will not do anything. So they will just pass by and not stop on them. But the moment when you add an antibody to the tumor cells, this is what is happening. So basically you see that the neutrophils are accumulating in the tumor site, okay? So, and this is one of the main mechanisms of tumor killing that is mediated by the monoclonal antibodies. All right. So this was for my PhD project, so I was happy about that, and I realized that innate immunity is something that is really powerful to us. So we normally, the immunologists, they always look at the adaptive immunity as if it is the most important thing that we have, but neutrophils are innate immune cells and are really powerful cells, and innate immune response is our first line of response, and it's really important because it's really powerful. So the next project that I was working on in let's say modern way, we can call it the lockdown project. Why I say that? Because, so if you have a virus, let's imagine that you have to be, let's say, fashionable. We take the coronavirus, okay? So this is the COVID-19. So we know that the coronavirus infects the first cell, okay? That we'll design it in black. So the first cell that is infected, of course, will be surrounded by neighboring cells that are still healthy. And the first cell, we know that has a really particular reaction. So first of all, it downregulates some molecules that are called MHC+1 molecules. Then, of course, it will downregulate the protein synthesis, okay, because the viruses use the ribosomal machinery in order to replicate. And third, this cell, what it will do, I'm sorry, I don't know what, this will produce interferon, okay? Interferon alpha. So this interferon that is widely used also in clinic, for example, in hepatitis, we use it a lot, it's a signaling molecule, okay? So it will signal to the neighboring cells that something is happening in the first cells. And the neighboring cells will respond exactly in the same way. So they will downregulate MC+1 molecules, I'm sorry, and they will downregulate the protein synthesis. So one of my main goals during this project was to understand, so from the first infected cell, so how much the so which is the diffusion, okay, of the interferon molecules. So to do this, I use a simple trick at the time. So basically, you can in vivo, in a mouse model, color in green the first infected cell, and then you can see in red the activated cells, so the cells that have perceived the signal. So, and to do that, I will show you some of the a really simple experiment that we did. So basically, this is a lymph node, and as you can see here, I will enlarge this image here. So this is an infected cell. This in blue are the neighboring dendritic cells, and in red you have the activated dendritic cells, basically the cells that have sensed the interferon coming from the first cell. So, and as you can see here, this first infected cell has all this cell activated here. So basically, I was interested in understanding how far this activation goes, and you can do it in a really simple manner, in the sense that you have this image. Basically, you can split it into channels, and if you make a really particular analysis that I'm not going into the details of it, you will understand that from the first infected cell, interferon runs up to 80 micrometers. So why 80 micrometers is a particularly important thing? Because you have to imagine that one cell is nearly 10 micrometer. So if a cells get infected, basically you have 8 other cells, okay, that will go into lockdown. Okay, and this is, guys, is particularly relevant for what we are living in the society right now, because so you see that all the country are actually shut down, but this has a really particularly biologically relevance, because if one cell shut down 8 neighboring cells, you can imagine that we have to do the same as human beings. Okay. So then I switch to the more, if you want, clinical side of things.
When you showed us the lymph node, Marcello, and the dendritic cells, is this all dendritic cell populations? So the DEC205 positive and negative, all the DC population in the lymph node, or is it
CD11C staining?
Okay, so a pan dendritic cell marker.
It's a pan CD, and the red one is the CD86 staining.
Okay, all right.
It's really easy. So basically, then I jumped back into the clinic. Okay, so, and I kept in mind that I was working with antibodies. Okay, and the antibodies that I'm focusing on are mainly IgE. Okay, IgE are important for allergy. Okay, I think you all studied that. And as a doctor, you know, if some of you will decide to be a clinician and a basic scientist, as I am, you will have to do some clinical project. And in particular, I am interested into drug allergy, venom allergy, and other things. But you can also apply your basic science to translational medicine, okay? So you can do what the dendritic cells are doing. So translating what is happening in the outside world into into your medical world. And I just give you a brief example, really in 1 minute, of an example of the project that we have been working on. So in allergy, if you have an allergic patient, one of the main treatment that you can use for example, this is an house dust mite allergic patient, you can perform an immunotherapy, which is basically giving, if this is the allergen, over time. So you give increasing doses of allergen over time. And at this point, that is called the maintenance phase, you will have fixed doses of allergen administered every 4 weeks. At this point, my patients were telling me, doctor, I don't know why, at the 4th week, I start to have the symptoms back. And again, to make a long story short, what we found out in our lab, and this is actually was my wife's work, that she also is an immunologist, is that if you look at T regulatory cells in this patient, so this is day 0, you perform immunotherapy, the Treg cells will go up, okay? But then, before the next administration of the immunotherapy, the Treg cells will go down, and if you perform another cycle of immunotherapy, the Treg cells will go up again, and then it will go down. So basically, we found out that this is one of the main mechanisms by which immunotherapy induces tolerance and induces cycles of Tregs expansion and contractions. Okay, so basically you inject the allergen, Tregs expands and then they decline, and then you inject the allergens, they will expand and then go down again. Okay, so this was I think I am on time. If I'm if not, just
no, you're in that time. This is, I mean, what you presented just now, Marcello, I've got a million questions about, but I don't want to take too much time because we want to keep on track with the webinar for today. Thank you for the very informative and very interesting presentation. You definitely have a knack for it, Marcello, and I'm looking forward to ask you about it after the end of the webinar. But for the sake of the webinar, and for the sake of our audience, let's start on a basic standpoint. Maybe a little bit less science and more of the recent events now in Italy. So we'll start by that, and then we'll walk into the science. So on a brief description on your side, can you tell us about the chronological story of what happened in Italy and where we are now, and then we'll take it through an immunological route thereafter.
I have this sort of mindset deviation. So I always bring things back to immunology. So Italy, I hope you guys will visit my country. Okay, it's a country, I will try to draw it in this way. Okay, so this is Italy. Okay, and if you look at it like this, it's more or less like an antibody. Okay. And this is particularly relevant for the situation in Italy that I'm going to tell you now, because, so I'm here, okay? Milan is here, Rome is here, okay? So Italy as a country is also functionally shared into different parts, because I think you all heard of it, but basically the north of Italy, and I don't have to say it loud because people from south of Italy, they don't like this, but it's still the economic engines of the country. And the south of Italy does a lot of the job, but the economic engine is at the north. So when it comes to the coronavirus, it's a really particular situation. So I will draw you a timeline here. And so let's start. So this is time, okay, in days. Okay, so this is what happened in our country. So we had a day 1, okay, where we had our first case. And the first case was discovered, and I will mark you in green, in this part of Italy, and it had a connection with another. So the patient that was discovered positive for coronavirus had a connection with a particular city that is called Lodi, okay, that is at the north of Italy. Then already by the next day, by the day 2, we had the first death from coronavirus. And right at this time, because so you have to know that European countries, so we were really watching what was happening in China, but I think at the time we were not thinking that it would come into our country so fast. So what happened that is immediately at day 4, our government created something that is called red zones. Okay, so zones that were closed, that were more or less here in this part of Italy. And what does a red zone mean? So red zone, so we will draw it in this way. So basically you have, and I can maybe write it with the typewriter, that it will be better. So you have no schools, okay, so the schools were shut down. Then you have, of course, no museums, no concerts, everything and was shut down. And the restaurant, for example, were open maximum until 6 p.m. Okay, and this was by day 4. Then, of course, the disease started to take place, but by day 6, I have the word in French, but the first citizen of Milan, the mayor of Milan, said, listen, this is not going to work, because the north of Italy, we are the economic engine of this country. We cannot shut down Milan. So basically, it ran a campaign that was called in this way. So the campaign was called Milano doesn't stop. Okay, so it doesn't, so no stop. Let's put it in this way. And this was really a huge mistake, okay, because everyone was going into the bars, the museum were open again, Il Duomo di Milano, that is one of our main church, was open back again to tourists. And to make, again, a long story short, not that long, but by day 18, at this point, so basically 12 days later, we have a lockdown, basically, of all this part of Italy that I'm driving you here. Okay, so they decided, listen, all the north of Italy will be under lockdown. And you have to know that for our legislation, and I think in your country might be the same, so you cannot say something and that thing that you're saying will be immediately applied. So it takes 24 hours, of course. And during that 24 hours, a lot of people fled, okay? So basically they ran from the north of Italy to the other regions, okay? And that caused, of course, a really scary thing, because if some of these people were infected by the coronaviruses, of course, at that point, they were spreading the infection all over our country. So at that point, our government, and this was by day 20, okay, our government decided to perform a complete lockdown. Complete lockdown, and this was since March the 9th. Okay, so, and we are still in lockdown, and we will be in lockdown until, so far, so this is our last news, until the May the 3rd. So, and to give you, because when probably you say it by word, you say, okay, listen, we are in lockdown, and you can say, okay, so what? So what changes? And an interesting thing is that some of scientists, and this is why science is really great, and I think I'm really passionate about it. So some scientists perform this study. So this, I can maybe show it in this way. So on the left, so this is Italy, this is my country, and on the left side, you see a normal country, okay? So in green, you have normal travel. In yellow, you have people that have traveled half of normal. And then in gray, you have no travel. And as you can see here, by the time that was passing by, the people were traveling less and less. And as you can see on the right side, on the very right side, nobody's traveling anymore. And this is really a nice thing for the pandemics, for the epidemics in general. But I can assure you that it's really difficult for us because, for example, I work in the south of Italy, but my family is from Rome, and of course I need to keep in touch with them. I would like to see them, but of course, for now, it is not possible. So, and to give you the last update about what the situation is in our country now. So if we have the, so if these are the number of people that have that are positive, okay. So basically, our curve is at this point now, okay. So we're starting to decrease the number of. So this is today, okay. It's starting to decrease, but it will take a bit sometimes to to have these restrictions ongoing. I don't know if that answers your question.
Yes, yes, it does, and it's a very significant point. If you can scroll down, please, down to that. So that point is the beginning of the ascend. You've met the plateau, and now you're going down. How long do you think that will last until you go down to you've got very little cases?
No, I think it's hard for me to say. But I don't think that. So zero is I don't know if zero is the main objective of this. So basically, we have to find a way to live with this virus. Okay. So basically, I don't know. If zero will be a possibility, but of course you can have a minimum rate of contagious, a minimum rate of positive patients, and that will be acceptable.
Do you mean by merging it with seasonal flu?
Of course, every infectious disease can have this kind of burden. I can have this kind of problems. But the main point of this infectious disease, and then we will talk about it later, is the number of people that needs to go to the hospital. Okay, so the possible consequence of it, because of course, if you, for example, take the either the HPV or other things, so the number of people that are actually positive to that, it's a lot, but this is not a problem for the healthcare system. This is a main issue for our healthcare system. So it comes to a point where it will be we will be at our limit. Okay.
Yes.
And I've heard, but this is really our last news from yesterday night in the newspapers, that they wanted to, for example, develop also some mobile phone apps where you can track the people movements and the positivity of the people and things like that, in order to be able to have a wider picture of how the infection was going.
Well, thank you for that brief background on the situation. And obviously we hope, in terms of clinical immunology, how would you describe COVID-19 from a clinical immunological stand? How would you describe it?
To tell you and to all the audience that, of course, everything that we say and everything that we're dealing with, it's an emerging disease. Okay, so it's something that is new. Okay, one of the main features in medicine and in general in science is that you have to fully understand the basic mechanisms of how the things work to be able to well define them. Okay, so I would, from an immunological point of view, for what is my knowledge of it, and by the way, I would like to tell everyone that for now, all the possible papers concerning coronaviruses are free in the literature. Okay, so my first observation in terms of that, and I got it here. Yeah, I saved it for you. So this was from March 2013. Okay, so this is the cover of Nature. Okay, and that was coronavirus targets. Okay, so this is not, coronavirus is not a new, a really brand new problem. It's just that now we realize what we are facing at. So from an immunological point of view, there's not much difference so far between, for example, SARS-CoV, okay, so let's call it CoV, that is the one that gives the first epidemic, and KAU-2. Okay, this is not that different because, of course, I think you've heard of it. So basically, this is a virus that has a single-stranded RNA, and it's important to state that it's a positive single-stranded RNA. That means that once that it comes into the cell, it can straight directly replicate. Okay, and then this is really important from a clinical point of view. So what defines this virus is that you have Hess protein, that are the spike proteins, then you have, of course, other proteins that will be the nuclear protein, the envelope protein, you have the hemagglutinin protein. But what, as far as it is for example, the KAU, so the first, not this one, but the one that was responsible for SARS, this protein, so the spike protein, was responsible for the immune response. Okay, so this was the antigen. Okay, so antigen, it goes with words. So basically, antigen is an antibody generator. Okay. Okay, so basically, if you immunized, of course, the people, if people get infected at some stage, if you look at the Ig titers, that might be, so you will have a spike at first in the IgM, and then slowly you will have a spike in the IgG. Okay, so what is the role of these antibodies, and what is the main problem, okay, during the infection? Is that basically, I think you know from the basic pathologies that viruses, in fact, are intracellular pathogens. So basically, the pathogens goes inside. So what is happening is that if you have an antibody that is either an IgG or an IgM, that will have this structure here, okay, that is a pentameric. So the only role that will have this antibody so far is a neutralization. Okay, and this is particularly important because in an active phase of the infection, you won't have the that much effect of the antibodies. What changes is instead having specific T lymphocyte that will kill, okay, the infected cells. And this is what is really peculiar of the cells, and maybe what you wanted to know. But is someone driving on the board, by the way? What is important is to have specific T lymphocytes to have a reaction in, to have a proper immune reaction. What we are seeing, for example, in these patients, and I think you all read about it, is a relative lymphopenia. Okay, and if you, for example, plot here, so basically you have different levels of it. So these are the number of lymphocytes, and this is the time. You will have patients that are relatively either asymptomatic or that are doing okay. Mushraf, I can't really viral infection. What is important, we all know, are the T cells. So if we just plot the number of lymphocytes, and this is quite peculiar for these patients, okay? So if we plot the number of lymphocytes against time, so what you will see, so these are the number of lymphocytes, is that the people that are actually doing okay will be here, okay? So this will be, let's say, okay, so these have less symptoms or that recovers well. So then you will have here people that will need to be hospitalized, okay? Let's call it AH. But what is interesting is that the people that have few lymphocytes, like the people that are here, so either they are in intensive care unit or unfortunately they die. So why is this happening? This has not been described so far, but you have to know that some of the studies have been done on the first KAU virus. So that is the one that gave SARS. So basically everyone is talking about this. So one of the main entry points of the KAU virus through a spike protein is that this receptor that is called ACE.
ACE2, yes.
ACE, but and this ACE, of course, will be expressed on different cells. So, such as, for example, lung cells, you will have an expression. The main point also is there is expression kidney. Okay, so you will have also kidney damage, and on the gut, for example, some of the people might develop diarrhea because of that.
We've seen also some papers on that, Marcello, that have come out recently, which discuss the distribution of ACE. It's widely spread. You see it in the lung, in the gut mainly in the small intestine, less than the colon. And then, ironically enough, in some of the immunohistochemistry where we stain only the tissue, they don't find any trace of it when they stain the tissue. However, papers have come out that show that ACE is expressed on T lymphocytes and macrophages as well.
That is actually true, and I had a slide on that. Okay, this is rhesus macaque. Okay, so this is non-human primate, but I think that can be applied also to humans. This is an immunohistochemistry expression of the ACE on all the different tissues, and of course it's expressed on the larynx, on the epiglottis, but in the same paper it goes straight also into the gut, and as you said, also on immune cells. But what I don't know if you discussed about it, and this was a Nature paper, so the COV has also another entry point that is called DPP4. That is the dipeptide peptidase 4. So this receptor is specially expressed also on lymphocytes. Okay, so the lymphocyte uses this receptor to in order to perform glucose metabolism and things like that. And so basically what you can imagine is that either the coronavirus might enter via the ACE receptor or via the DPP4 receptor.
And we do know that the new coronavirus goes through ACE too, as I was trying to comment on your point. I was going to ask Marcello, in our discussion yesterday with Rodney Rosalia, we now see some new research. Do you think that can attribute to what we see in the lymphocytopenia? Or is there a different mechanism that brings lymphocytopenia down?
So what I can tell you is that, for example, the infection of the coronavirus one, so you got the T cell and then you got the COV. So this infection is an abortive one. Okay, so the virus may infect the T cells, but it can't replicate into the T cells. Okay?
Is there a reason for that? Do we know why?
No, this has not been described, but what you can imagine is that for SARS-CoV-2, for some reason, it might drive apoptosis of T cells. Okay, so but this has yet to be described.
Yes,
that might be at the basis of the lymphocytopenia that you see. Okay, and you have to know that, for example, DPP4 or AC2 are, of course, they will have SNPs, they will have polymorphisms among the population, and of course, one thing that one can imagine is that a particular polymorphism of the DPP4 or ACE2 might grant the enter of the COVID-19 into the cells more than another polymorphism.
So you're attributing then the drop in lymphocyte counts and the increase of WBCs, or focusly on lymphocyte counts?
Yeah, I think it might be one of the mechanisms. You know, it's especially in the marked lymphocytopenia. Then there's another thing that the studies are missing at this point on this. So basically, you have also to define which is the age frame that you're looking at, because of course you will have some people that will have less lymphocytes because of other,
So this is another point we were discussing in type of TH you expect with viral infection is a TH1 immune response. Now this is evident with the increase in interferons, especially interferon gamma in the adaptive immune response. Now, of course, the timing of interferon gamma plays a critical effect, especially for somebody in the clinic where you see people, where you divide patients between ICU and non-ICU. In terms of the type of immune response you see between ICU patients and non-ICU patients, so if we can say mild and severe, what is the difference in the immune milieu
normally, I think, I don't know if you discussed this already, but aside, of course, a drop in the lymphocyte count, what you have in this patient is a high CRP, there is a C-reactive protein, and you have a lot of albumin, okay? You have to know that these are, for us, for clinicians, are not that specific markers of infections, okay? So it's just a marker that you have an infection ongoing. Then what is important is this particular thing here, that is the D-dimers. Okay, I don't know if you discussed about that, but
so it's a coagulation factor, I believe.
Yeah, so these coagulation factors have a really high risk. So basically, patients might develop for an unknown mechanism. So basically, you can imagine that you will have autoantibodies, for example, other infectious diseases. For example, if you take syphilis, syphilis will give you autoantibodies that will promote the clot formation. Okay, but we don't know about this virus yet, what is happening, but you have basically, in some of the patients, the D-dimers that are like going up, and this is really a risk factor because, of course, you will have intravascular coagulation, and this will be, of course, reserved to the more serious patient, if you want. You have also the possibility, and I think how do you say that, to dose the IL-6. Okay, so, and this, I think it's, it does not give you an indication on how serious a patient it is, but it's a good candidate for a treatment or for another, as you can imagine, you know?
Yes, it's definitely associated with severity, even though there still appears to be a part of discussion whether it's in the plasma or cellular, on a cellular level expression. Can I ask more about D-dimers? Because it's been coming up in many researches, if not one of the early researches that came out of China, where with Giuseppe Lippi, a fellow Italian of yours, what do coagulation factors tell us about the infection? So
normally I have to tell you, so in terms of clinically wise, so the D-dimers are normally asked to someone to exclude that you have a thrombotic event. Okay, so this is why as a clinician we use it. So I would rather say that D-dimers, and this is, and I'm gonna tell you why I'm saying this. So D-dimers are a sign that the immune system is getting activated, because, for example, if you take patients with, and this will see us in our clinic, so if you take patients with chronic urticaria, okay, there is a condition where you have itchy wheals, and some of the patients have a severe chronic urticaria that have, and they need eventually monoclonal antibody treatment. In some of these patients, you will see the dimers that are going up. Okay, so basically what we have to keep in mind is that so our immune system in our body is not you know, sealed compartment. So the complement system is really connected also to the coagulation system, to the coagulation part, and that, you know, makes an interplay. I would not say that it's a sign of tissue damage, but of course it's a sign of an ongoing infection that has reached a certain extent. Okay, I don't know if that answers your question.
Yes, it does. It does answer the question. I would like also to lead on a molecule. I don't know if you can help us with procalcitonin, which is also a molecule that appears in ICU patients.
But procalcitonin is not that again, like CRP. These are standard, I would rather say, molecules that we test for the ongoing inflammatory state.
And would you see that, like procalcitonin, like D-dimers, would you say they are solely indicators of inflammation, not indicating the severity of the disease? Is that fair to say?
So this, I don't have enough studies for this disease to tell you. So you're meaning, so the highest the level of procalcitonin, the worse it is.
So yes, the reason I'm asking about this, Marcello, because again, some of the early papers, like Giuseppe Lippi, like some of the papers from Habrel et al., they indicate it is an interesting increase where procalcitonin conventionally or is associated with bacterial infections. So,
Yeah.
So an increase in a bacterial infection marker in a viral infection suggests that there's susceptibility in generating an immune response, or as a secondary infection overcoming now. So might be a sign of the deterioration of the condition.
Yeah, I think, yeah, I know this, and I think it's the data's really, I can't answer you properly on this question because we don't have enough data on this. And on top of that, so for example, bacterial infection will, immunologically wise, will trigger, for example, if you take a gram-positive bacteria, of course, you will have a trigger, a kick on, for example, the like receptor 4. That is one of the main kicks of infection. You know, is responsible, for example, also of shock, of septic shock. And as for the viruses, I don't know. I think that, you know, for this, we would need correlation studies, for example, with severity of disease, procalcitonin. And then it's also important to, when it comes to symptoms, to understand that this kind of values in our bodies are not stable. So basically, you have a time frame in which you can make blood sampling. So I don't know if, for example, in an early stage of the disease or in a late stage of the disease, you might have differences that might influence your outcome.
Okay. Can I ask you about the serology? So the IgGs. The conventional wisdom now suggests that you have a delayed, or as you'd expect from IgG response, you have IgM and IgG developing between 3 to 9 days post-infection. How much are as far as we know so far, how much are antibodies involved in fighting coronavirus?
So as I was telling you before, I think that it's we have to think it in a larger way. I think that the being an intracellular pathogen, of course, the cell-based response will be more. Of course, you will have here an infected cell. This will lead to viral particle outside, and so the T cell can actually kill this cell that has been infected, but the antibodies can neutralize the virus here.
100%. You see that also. You see that with the type of T cells that are involved. You see an accumulation of CD8s, especially activated CD8, CD38, and the HLA. Would you say that the increase in CD8 is a good sign, especially the activated CD8, the HLA, DR, CD8 between day 7 and 20? Is that a good sign for the infection to have an increased?
I would say so. I would say so. Yeah. Because, you know, it's an intracellular pathogen. Okay. Then when it comes to lungs, okay, so we have also have to, I think, set the things in a physiological manner. So you have some organs in our bodies that have more compliance than others. So for example, the liver. So the liver first, so you can have an infection here, but the rest of it will be okay and will be able to supply the what you need. Of course, the lungs, as you might know, you have the bronchi, and then you go here, and then you got the pneumocyte here. Mm-hmm. So basically here is where the oxygen is changed, okay? And so if the coronavirus is, but this is true for nearly all the viruses that can cause pneumonia. So basically, if you hit this part of the bronchi, of course, here the oxygenation is not working anymore. You have a shunt effect, and this tissue has not that much compliance compared to other tissues. Okay, and on top of that, if you reason anatomically wise at the lungs, so if these are an horrible design of the lungs, but in the lungs, what you have here is the pleura. I don't know how to say it in English, but it's like this. And between this, there's a negative pressure. Okay, so once that you start to have problems here and accumulation of liquid here, this at the end might cause a collapse of the structure. okay? Because what keeps the lungs expanded is the negative pressure that is here. So basically, I think that it's a nice sign to have CD8 T cells because it's a sign of infection. But of course, a tissue damage and the capacity of a particular tissue to comply to the damage and a repairing system is different. And of course, when it comes to lungs, it's a little bit less, for example, than liver.
What markers do we have for the accumulation, the negative pressure falling into the alveoli? Some papers talk about, if I want to pronounce it well, but it's hyaluron, which is which leads to the accumulation of fluids. I don't know. Are you familiar with the certain?
Yeah, I mean, this you can see it in an X-ray also. But so the first thing that distinguishes that you can make, for example, that you can understand that something is ongoing at this level here is hemogas. I don't know if it's written in this way in English, but basically you, from an artery, you monitor the tension of O2, the tension of CO2, and the pH, okay? So in this patient, what is happening, and it's really particular, is that, as I told you, so this part of the respiratory system is compromised. So it's the part that is exchanging oxygen. Okay, so this will be nearly in some patients to 50% in the symptomatic patient, and this is really low because it's supposed to be 90, 99%, 98%. And the CO2, it's, you would say, okay, if I don't have sharing of oxygen with the CO2, this will go up, but this is actually going down, the CO2. Why is that? Because the patient is hyperventilating. Okay, so you got the rest of the lungs and the patient are starting to hyperventilate, and this will promote a reduction in the CO2. So just based on this simple analysis of the blood sampling, you can actually detect that something is happening at this point of the bronchial, and the pH is normal. Okay, you don't have variation in the pH.
And would you say this is kind of a natural reason why elderly people are more susceptible to the disease due to the weaker lungs, by better clinical terms, than the young?
So when you asked me about the elderly people, I really thought about that. So it's I don't know, I'm an immunologist, so I tend to focus the things on the immune system. So as we know, and as you know, how it works. So basically, if you got our virus, you will have first an innate immune response. You can have a resolution that is not the case, and then you can, with an adaptive immune response, you can fight the virus. So I think and this process normally, as you said, normally it takes 7 days, but for example, also 8 to 9 days when it comes to the coronavirus. So this, I think that we have to keep in mind how, and there are a lot of studies on how the immune system works in old people. And why I say that, because if I might, so for example, we have in our bodies primary lymphoid organs. Okay, so if we take, so I draw it in this way. So this is a tiny guy and this is an old guy. Okay, so this is young and this is old. So for example, if we look at the bone marrow of the young guy, you will have lymphoid cells and you will have myeloid cells. Okay, myeloid, let's say, cells that then will give rise to cells of the myeloid compartment. What is interesting is that in an old people, already in the primary lymphoid organs, so in the bone marrow, the precursors are less. And on top of that, you have more cells of the myeloid compartment. And it's not trivial. This is just because, for example, in the elderly, what you see clinically wise, and you can ask also to the hematologist, for example, you have the disorders like essential thrombocythemia, or myelodysplasia, that are basically due to, there are neoplastic conditions that are due to expansion of the cells of the myeloid compartment. Yeah, so then of course we all study that from the basic immunology. Of course, if we look at the thymus of a young adult, of course you will have a lot of cells that are the thymocytes that will have positive selection, negative selection, and this will give you a pool, okay, of proper T cells. But if you take the thymus of an aged patient, first of all, you will have less thymocytes, okay, and then you will have an increase in the adipose tissue, okay. So basically, the function of the thymus is declining over the years, and this might be a simple point, but if you have an aggressive infection on the lung compartment, and as I showed you, so the lung compartment have no compliance or less compliance compared to other organs. Of course, if you have less clones generated, you will have less clones that will recognize the spike proteins, and this will take you more time to generate a specific immune response. And during that time, your virus is making millions, okay? It's making damage to the respiratory tract. That is a tract, again, that has not that much compliance, if you want. And so I think that it's the point of the elderly. It's a balance between the how aggressive the virus is on the epithelial cells, on the pneumocytes, and on all our cells, and how much efficient T cells clone, and how much an effective immune response you can generate. So there
has to be a role for memory cells in here, Marcello. I mean, when I think of it personally, and again, this has not been discussed. This is my knowledge of immunology, so my very few years of understanding immunology. You need to have a familiarity with the phenotype of the virus to generate a good enough response to fight it. So if the elderly fail to produce a lot of immune cells, specifically the imported immune cells like the CD8, the CD8 HLA-DR activated cells, you'd expect that they have a population of cells that are familiar or have seen similar viruses.
So this is really an interesting point, and because I think you raised a really good point, because so this COVID-19, so we don't have evidence, of course, as you said, for memory T cells in this disease, okay, but we can discuss a little bit on how it works, for example, for the others. So this COVID-19 belongs to the beta coronaviruses, okay? So the beta coronaviruses is a family of viruses. Let's make it in this way. It's a genus of viruses that includes, for example, the MERS, that by the way has been discovered in your university, the SARS one, but you have also the KAU, and there are a lot of viruses, for example, that are the common cold, if you want, that do not have this kind of symptoms. So if we look, for example, and this there's a really nice paper, if you want, I can send the students the reference. So if we look at the protein, for example, the spike protein, the N protein, the envelope protein, and the hemagglutinin protein. So basically we can see the sequence homology based on the genome of the different proteins. So for example, if you consider the COV, okay, these are the main ones that have been studied. So the spike protein, I'm reading because I noted them, is 76%, and this will be 90%. This again will be 90% of sequence homology, and this will be 97.4%. So this is a lot, okay? So, but for the spike protein, that is the main of our immune response, as you see, it drops, okay? So I don't know if this is enough, because this is a sequence homology, and of course you will have other variation after the translation of the protein.
Yes.
But
and we don't know the nature of that protein as well. Is it convalescent, or is it linear? How is it presented on the surface? There are many questions related to that protein, other than the homology.
But what is interesting is that, for example, for the Middle East one, so for the MERS, if you see, and this is the same family, okay, this is the same beta coronavirus. For example, this drops up to 29%, this 45%, this will be 39%, and this will be 34%. Okay, so as you were pointing out, so I can imagine that someone that is an adult may have encountered a, let's say, a cold coronavirus, okay? So it's it catch cold that was due to a coronavirus that is not that aggressive, okay? So this individual might be protected. But as you can see, there's a great variability among the same family of these beta coronaviruses for these proteins. So it's, of course, you can imagine that there's a role of memory cells, but I think that we need data on the similarities between the structural proteins and the antigens to be able to
100%. 100%. I think the other thing that people, because this is a question my students ask all the time, why don't, if we know the homology, if we think there's a homology, why do we fail to mount a proper immune response?
Sequence homology is not...
it's not 100%.
It's not 100%. Then you have the folding problem.
Folding, exactly. So recently in Kuwait, I saw yesterday in the news, and some of the attendees might have seen this, they were asking patients who have been cured from COVID-19 to donate blood for plasma purification. I'm thinking, from what the news title was suggesting, this is just mainstream media, they were suggesting that maybe some isolation of IgGs to help them re-enter or to re-administer into sick patients to help them cure. So that brings me back to the question of the B cell. Can I ask you one more question? In terms of the immune response, Marcello, is it different between these different beta coronaviruses? Do we see a different mounting of immune response, or is it all a CD8 natural killer cell, CD4, interferon gamma immune response?
So I don't know. I don't think that there's any difference in how the immune response is mounted. And what I can tell you that there is a very recent paper on, I think it was Nature. It's really last week, I think. So basically, if you take, and it was all, of course, I'm interested in immune cells. So if you take lymphocyte cell lines and you make them express the entry site, okay, that is this one. And then let's say it in this way. So this is COV1 and this will be COV2. So what you see is that for some reason, so this will be 1 and this will be 2, and this is the percentage of infection. What you see is that the SARS-CoV-2, so that is this one, is really more aggressive on immune cells compared to the COV1. For some reason, okay. So this is really a simple experiment where they have challenged, make the cell line express this receptor, and then they challenged with the virus. And what they've seen is that it's really more aggressive. So I think that it's not, again, a matter of on how the immune response is mounted. And of course, then the chapter of the cytokine storm, it's another huge chapter, because it brings us back also to the flu pandemics of 1980. But again, this is not on how an adaptive immune response is mounted. I think that the cytokine storm might also be to an innate immune cells. Okay, so it might be to toll-like receptors, polymorphism. It might be to simulation of toll-like receptor and things like that.
Well, we were mentioning that on a different note as well, how we believe toll-like receptor 3, toll-like receptor 7. You've got nods and reg-like receptors that are responsible for detecting intracellular infections like viral infections, especially RNA viral infections, as we see with corona. I would like to go to treatments. Now, we've heard treatments everywhere, and I'd like to ask you specifically to start with, because this question has been thrown in many times. So, a paper out of France, I'd love to see the name right, Philippe Gautret, which is one of the first papers out of that kind of treated unspecific group. They don't talk much about the group they've treated or their age with hydroxychloroquine and azithromycin. And they've shown very promising results for hydroxychloroquine when associated with azithromycin. Can you tell us a little bit about hydroxychloroquine, azithromycin, and has it been tried in Italy?
So let me start from the second question. So yes, the answer is yes. So if you have a COVID-19 patient, basically what you have is that the treatment that is given now is, as you say, hydroxychloroquine. Let's make it in this way, and the dosage is 400 milligram bis in die, okay, 2 times per day for the first days, and then you switch to 200 milligrams bis in die, okay? And the second treatment, and this is done, okay, but these are 2 different aspects of azithromycin. You started using 500 milligram, okay, per day. But normally, so the treatment with azithromycin is supposed to be 3 or 4 days, and then here you make it last longer, so at least 7 days. Okay, so these are how the things in clinics are working now. So this treatment, and as I told you, I've also asked my colleagues in the that are dealing also with more severe patients, and this treatment is actually ongoing. Okay, and you have to imagine that in this kind of patient, of course, you will need oxygen supply and other things. Okay, so
is it only severe patients, or even when is it administered? When do you give the hydroxychloroquine? Is it early in the infection, or when they're administered first, or ICU patients?
I can tell you that, for example, in the hospital, so we hospitalize patients that are moderate to severe. Okay, so it's the one that is positive that for now stays at home. Okay, so probably this information is missing, and I think it's important. So you will have, let's say, asymptomatic or pouchy symptomatic will stay home, okay, for 14 days. And then the moderate to severe are actually hospitalized. Okay, why you are hospitalized? So the parameters is the fever. So basically, if you have fever, nearly 38 degrees centigrade, if you have cough, if you have dyspnea, in this moment, probably this is due to COVID. Okay, so this goes straight into our COVID unit. Okay, so and in this patient that has been hospitalized, you start to perform this treatment. Okay, so upon admission, they start with hydroxychloroquine or azithromycin. Okay, I think that the macrolide point and the azithromycin point is the one that I can develop a little bit more, because it's you know, if we try to look at it in a wider way, so. If you look, for example, in chronic asthmatic patients, and there are several groups that have studied that for some reason, okay, the mechanisms behind it is quietly unknown. For some reason, in chronic respiratory inflammation, if you use macrolides, if you use azithromycin, what's happening is that the patients are doing better. Okay. And so it's not the first time that in lung disease or in pneumology, azithromycin is used. Okay, I never use it myself for our chronic asthmatic patients, but it's not the first time that this has been done.
And it's been used preferably in Ebola and the Zika virus. So azithromycin has a good track record, we can say.
Yeah, it's a good track record, but not only, what I would like to say is not only in infectious diseases. Okay, so also, so if you have chronic asthma is a chronic inflammatory disease. Also in inflammatory disease in general, azithromycin for some reason does better.
What does it do? Do you know the mechanism behind it?
I can't tell you in the detail the mechanism of azithromycin. Okay, so it's because it's more well characterized, the one with hydroxychloroquine. Okay, it's hydroxychloroquine, of course, as you know, it's been developed as a malarial infection preventive drug. So, okay, so basically it lowers the the pH of the lysosome, probably so it does a better cell killing, because, for example, and this doesn't work for all the viruses, for example, if you have a virus here, it's sensed by the TLR, and it can go into the lysosome. Okay, so pH. For example, this has not been described for the coronavirus, but for example, if you take rabies viruses, okay, I used rabies viruses. So rabies viruses basically can resist to the pH that is in the lysosome because they change the structure of their surface proteins. Okay, so once that they get into the lysosome, they fold, and then once that the pH is higher, then they open back again. Okay, so probably hydroxychloroquine is working because there's an issue of the viral killing inside the cells, but this drug, as you might know, has also been used, for example, in the treatment of some rheumatic diseases, such as, for example, arthroid rheumatitis or things like that, as an immune modulator. We are in a framework of medicine that is, antibodies, monoclonal antibodies are really expensive. Okay?
Yes.
So giving them on a large scale is really difficult, but I think that one of the main advantage, and it's working, okay, so this kind of treatment is working. One of the main advantage, and I think we have to keep it in mind in medicine, is that if you act on a system, but this is true for humans, for cars, for objects in general. So if you're trying to modulate a parameter, you have to be able to measure it. So I think that one of the main advantages of tocilizumab and of the anti-IL-6 antibodies is that you can actually measure what you're looking for. Okay, so you can make pre and you can make post, and you can eventually adapt the treatment. With hydroxychloroquine, you can't do that. But these treatments are actually used for the treatment of COVID patients. But an important point, for example, for IL-6, is that you might have heard of it. So one of the things that you can use is steroids, okay? And steroid and IL-6 in the treatment of the patient with COVID-19 have that's an important point to keep in mind. So if you give a steroid to a patient that has really an ongoing infection, you might do worse. So basically, in all these patients, you have to measure the viral load. Okay.
Prior to treatment.
Yeah, so once that the viral load is starting to diminish, you can give anti-IL-6 and steroids, because otherwise, it might be a little bit dangerous. This is not happening.
This is very interesting, Marcello, because I've seen the publications that suggest that corticosteroids are detrimental, but nobody seems to say why. Do you have an idea?
Well, I think that
because you are seeing a pro-inflammatory immune response that is harming the patients. You'd expect corticosteroids to help, however, they're not. Is there something to do? Is it more with the mechanism of corticosteroids, or something in the environment of the tissue?
I think more, but again, so this is just a discussion between me and you. Of course, you need experimental data. So I think, again, we have to look at it in a broader way to our human body. So the steroid receptors are expressed everywhere, okay? So as I told you, one of the main mechanisms that prevents the viral replication in other cells is that you have a communication between cells, and cells get activated and say, okay, something is not right. Of course, steroids will abolish this, okay, in every tissue, okay, they will do this, because of course, it's an anti-inflammatory, okay? So basically, you shut down the inflammation, but of course, the balance between where the inflammation is good and where the inflammation is harmful is quite difficult to say it now. But we have to keep in mind that, for example, the IL-6 receptor is not broadly expressed, but every cell of our body has steroid receptors. Okay, so basically, if you give dexamethasone, that is the one that is used now, what is happening is that basically you're acting on all the cells of the bodies, including the pneumocytes, including, you know, muscular cells and things like that.
So an unspecified effect.
And there was another point that you raised that I think is important, and that in our hospital they are starting to do so. And apparently it's working quite fine, but I don't have literature to give you. So you have a sick patient and you have a patient that have recovered. So, and what they're trying to do is to serum, okay? So believing the serum transfer, believing that you have, of course, IgG, and believing that you have IgM, that will protect this patient, okay? So this is something that in our hospital is done. It's not yet published. It's not, but as you were saying before, so basically antibodies can also play a role in neutralizing the virus, and you can transfer these antibodies, okay?
I'm going to add the point, Marcello. The dose you guys use for hydroxychloroquine is quite lower than what these papers have been suggesting. I think you said 300 milligrams, if I'm not mistaken.
No, it's 400 milligrams 2 times per day. That makes
2 times per day.
Yeah.
So they use 600 milligrams once a day.
No, so it's I'm sorry. So it's 400 so it's 800 milligrams twice per day. So you make a charge of it, and then you switch to 200 milligrams twice per day. It's so it's 400 milligrams, and you have to consider that. I mean, you have different protocols, but every drug has its own. I don't know how to say it in English. It's half-life.
It appears as azithromycin is there to protect from secondary infections because it's an antibacterial antibody. Is that fair to say that you present azithromycin with hydroxychloroquine to prevent secondary infections, possibly in the lung?
No, I mean this is of course yes. I mean it's, but it's not just restricted to COVID infection. You know, I mean once a patient hospitalized for a pneumonia that you can't say that is viral, of course you, in order to prevent secondary infection, you can make an antibiotic treatment aside that.
Yes. What about remdesivir? I don't know if you've used the antiviral. Some papers by Wen Cheng Ku.
There have been contrast opinion about that. So at the very beginning of the epidemic here, they started to use it, and then in our hospital. So these are really real life data that I'm giving to you. Okay, so I think it's also important for you to know what in real life is happening. So at the beginning, we were giving there is an association between 2 different proteases inhibitors used in the HIV. So that is lopinavir, ritonavir, I believe. But then there has been some contrast in the literature, okay, so about the effect and about the potential efficacy of it. So for now, we don't do it anymore.
Is there a reason for that?
I think it was just based on the literature, okay, and then we're not seeing that improvement on the clinical point of view.
And you were talking about anti-IL-6 treatments. Now we know IL-6 is a drive for inflammation. It plays, it orchestrates many roles. Specifically, or more likely, the T helper 17 milieu of immune responses. Have you guys tried anti-IL-6 blockers without steroids? Without the steroids? So alone anti-IL-6?
Yeah, I mean it's either one or the other. You don't do it. Yeah, no. I mean, I was probably unclear. It's just you see this coupled here because in both of them you have to check the viral load.
Check the viral load. Can I ask about any other immune mediators that that the hospital has tried to block? TNF alpha, interferon gamma blockers.
Yeah.
Any of them? Because I know they are the first candidates in any pro-inflammatory disease, any disease that might lead to multiple organ failure. Has anything worked with that?
Are you asking for my personal opinion, or?
I'm asking if you've tried it in the hospital.
No, we didn't try it. I know that they want to know. So these are the treatment that we have tried. So hydroxychloroquine, azithromycin, the antiviral, anti-IL-6, and corticosteroids. Okay. And I know from a friend of mine that is working closely with this patient that another drug that they wanted to try is this one is called tofacitinib. That is basically an inhibitory of Janus kinase. And I think that makes sense to try this one because as you know, Janus JAK-STAT signaling pathways. So basically, if you get, you mentioned interferon alpha, okay? So every cell has its own interferon receptor on the surface, and JAK-STAT, it drives into the nuclei then the production of other cytokines. And I can show you, I was working briefly on that. So I think that this particular treatment makes sense because, so for example, this is a simple experiment where you see STAT in green and the nuclei in red. So basically, if on the cells you throw interferon alpha and interferon beta, what you see is a translocation of STAT from the cytoplasm to the inside of the cells. And of course, then you can quantify it. But so to have this target is not that bad because it's, you know, you can, let's say, skip the blocking of interferon and you go straight directly.
JAK. Well, anti-JAK3 is an inhibitor of T cell functions, so it does make sense to work on JAK. I don't know how much it's used, to be honest. I'd love to see.
This is really the beginning of it. Okay,
beginning
so
of use.
yeah, and then the same applies to the what we said for the steroid treatment because JAK-STAT, it's an important adapter for a broad panel of
cytokines and cells, of course.
First, you will block everything.
Marcello, let's take some questions. What do you think?
Well, of course.
Ayat, are you there, Ayat? I know she's been listening intently.
Ayat, did I made you sleep?
No, I'm just trying to. Ayat, can you hear us?
Yes, I can hear you now. Thank you for the talk. It was very exciting. And of course, I have lots and lots of questions, and even the audience here, they have so many questions. So going back to using of the chloroquine, I know that one of the lessons that we learned when using the chloroquine with malaria, that the Plasmodium parasites develop resistance to the chloroquine because of the overuse of the chloroquine. Do you think we might face the same thing with the overuse of chloroquine to treat the COVID-19 patients?
So honestly, I don't think that, because for example, if you compare, so bacteria, of course, they adapt also to, for example, bacteria and parasites. Okay, they adapt to, for example, also antibiotics if you think about bacteria. But the viruses is quite different. It's quite different because, for example, if you consider HIV, so one of the main problems of HIV is that it makes errors, okay, in the reverse transcriptase. So that guarantees a mutation of its phenotype a long time. So the main point of the coronaviruses is that they don't make errors. Okay, so let's say the shift in the phenotype is not, at least according to the literature, is not that common, but it takes time. So for example, I don't know if it helps, but for example, we all know that the coronavirus started from the bats, and over time that there has been a shift mutation that brought him to an intermediate animal. Okay, that I don't know if it was a lemur or another one, and then it went into humans. Okay, so, and this mutation goes on in this direction, it cannot go back in this way. So it's, I think that they are pretty stable as a genome because they don't make mistakes in terms of replication. But then, of course, this is an immunology sport, and
let's say immunologists hope
Yeah.
Ayat, I'll come back to you in a second. Let's open. Let's see some of the people who raised their hand, if that's okay with you, Ayat.
Sure.
There's Ghaida. I'd like to see if she can hear us. Ghaida, hello.
Hello.
Hi.
Assalamu alaikum.
Alaikum salam. What's your question?
Great lecture and beautiful drawing, mashallah. My question, what do you think about herd immunity? It's good idea or not?
Very good question.
Why?
Very good question. Thank you, Ghaida.
Awesome.
Welcome.
So herd immunity, but again, this is my own opinion. So we can, it's fun that you've asked, because it's, if I can take some time to answer you, it's, I've made a short video also for Italian people about that. So if you have, if you take the infection, okay, so if in red we take the virus, but this is true for all the viruses, okay? So this is the virus, okay? And then you will start to have an immune response, okay? And this, of course, you will have this part of the curve here that corresponds clinically wise to your symptoms, okay? So if you translate this into the society where we are living now, what is happening is that herd immunity function exactly in the same way. So basically you have sick people, okay, that then will start to drop. And I showed you that in Italy, we are more or less here. And what is changing over time is that you will have, let's say, an immune response, but these are actually people that are developing immunity. And of course, you will have this part of the curve that in a society manner will be the symptoms. Okay, and this is what you're reading in the newspaper. So basically, in the hospital, you have a lot of people. In the intensive care unit, you have a lot of people. But why this is working? So this kind of principle. So one of the mechanisms by which society-wise herd immunity might work is that, so if you consider this as a sick individual, okay, this nice drawing here. If you put this individual close to a happy one, an healthy one, what will give you is two sick people, okay? Because this will be, of course, contaminated. But what is interesting about society and about this work center, this is the work of people that are working in the hygiene department in our country and everywhere. So if you have a sick person that is surrounded by someone who is happy but is immune, okay, so that has developed mechanism of defense that we still have to really define for this COVID, this will be for you a sort of a shield for the transmission of the infection. So basically you will always have all your people happy here, and then you will have just this one to take care of. And this is really even more important when it comes to this pandemic, because just one sick person can infect 3 different ones. Okay, so you can imagine the extent of it on a large scale. I don't know if that
are we depending too much? Are we relying too much on the immune system in herd immunity? Is it unfair, you think?
I don't think it's unfair. I think it's, if you want to have my own opinion, so all the living beings have an immune system. Okay, plants have their own immune system, and we have an immune system. So ideally, if we are more developed, okay, we have memory and we have specific immunity, but if we would be evolutionary that advanced to have an immune system able to fight all the possible pathogens, tumors, and etc. of the medicine that we know, really there will be not much to say. Probably traumatology, genetic diseases, these are the only things that would still stand. So I think that the immune system is a great part, but I think that the mistake that we make is that we think that since we are primates and we are smart, that also our immune system is that developed. I don't think so.
I think it's relying too much. And there's one more factor that I don't understand why nobody takes into account, Marcello. is the demography. If the majority of my population is older, what herd immunity am I waiting for? To wipe out 20% of my population or 30% and then wait for the remainder to develop herd immunity? It doesn't make sense in that front. I'm not an environmental scientist, so I don't know, but I know my immunology. And from that, I don't understand how you can depend on herd immunity in every and any population. It doesn't make sense. Ayat, oh, sorry, you were saying, Marcello.
No, it makes sense. And then you have to add to that is that, so pharmacology-wise, we don't have that many treatments available for viruses, first of all. Second, in terms of demography and the way we live, so basically in response to a pandemic, or the only option that you have is the lockdown. But what has changed over time is that we travel faster, okay? So the disease spread faster. So our means are always the same. So we don't have antiviral, we have to lock down, but the virus takes advantage of our own, society and how do we work.
Yes, 100%. Ayat, a question from the audience or from yourself.
Can I just ask one question when you just talked about the transmission? So we know that the virus was also isolated from the feces. So do you think there could be like a fecal oral transmission route?
That's a very good question, because I've seen one of the papers, or King et al., one of the first to show that you've got viral RNA in the stool around day 7.
Especially that, I know it's rare, but they still reported that the patient might develop diarrhea. Again, this can give another hint.
I don't know if it's rare.
Sorry, develop what? Sorry, I missed the word.
Diarrhea.
No, it's you're totally right. I mean, but that is the function also of the expression pattern of the, as we were saying before, of the entry receptors. Okay, so they are expressed in the intestine, they are expressed in the gut. So the virus can affect the gut. So of course, when it comes, as you say, to the transmission, of course, you can have this kind of transmission that is typical, by the way, for example, E. coli is transmitted in this way. Okay, so it's you're completely right to think that. And I add on top of that an observation that I think that the kind of immune response that we make, it's conditioned by the organ where we have the damage. Of course, for example, in the gut, you would see a lot of IgA response. Okay, so the final clinical outcome of an infection might also depend where the entry site is. Okay, so basically, if you have something developing in the gut, probably your immune response won't be the same. Okay, because the immune system is told what to do by the tissue. And this has been a lot of work by really renowned immunologist that is called Polly Matzinger, that was working on NIH about that.
I want to add an important point here, because it appears what you're suggesting here, Marcello, is very important. So ACE2 has been expressed, now this is by immunohistochemistry, so staining. I'll bring the name of the journal and the paper in a few seconds. Alveolar epithelial cells, type 1 and type 2, enterocytes all over the small intestine, but not the colon. In the arterial and venous endothelium in all organs, arterial smooth muscles in many organs, and myofibroblasts and fat cells even. Now, it's important to note that this paper did not identify them on immune cells. However, we have other publications that do, and this is by Ing Hamming in the Journal of Pathology, 2004. So as you were saying, the entry sites are vastly expressed all around the body. Mm-hmm. I have a friend of yourself and mine, the lovely Antonella Farah. She has a question. Hi. Antonella.
Ciao, Marcello. Great to see you. Thanks for your presentation. Hello, everyone. I have 2 questions. Sorry, I joined late, so I don't know if I missed this. But I wanted to know what's the situation in Bari. Are there a lot of cases? And then my second question is about the antibodies. So protective antibodies, there is a lot of confusion. It is not clear yet whether patients do develop protective antibodies. Also, some tests are circulating and some people started warning they're not reliable at all. I think they are very important because they could offer a way in which we can gradually start reopening after this long lockdown. But it is very confusing. Some people report that the plasma can be used to treat other patients because it contains the antibody, but some others have reported that these antibodies are not effective or not present, which is very scary. Thank you.
So no, thank you for the question. So in Bari, that is the south of Italy, I have to say, I'm glad to say that the situation is relatively under control. So of course we have cases, and we had a scary moment when a lot of people fled from the north of Italy to the south before the, let's say, partial lockdown of our country, and we were expecting an outbreak at the time, but this actually never happened. And so we managed to control the situation for now, but with a lot of efforts, in the sense that, for example, an entire building of our hospital, that is one of the main hospitals of south of Italy, that is a university hospital, has been just dedicated to that. And this, of course, has shrinked other services. So, for example, the surgery services and the even for example, the clinical allergy and immunology center is shrinked, and we are just dealing with emergencies and with desensitization, for example, for oncologic patients. But with this kind of measures, I have to say that the situation is still under control. So as for the second question, as far as I know, yeah, the antibodies. I think that there's a lot of, as usual, there's a lot of confusion on, I think that there's two different questions. So the antibodies and the detection things. So first of all, I don't think that IgG and IgM have the same ability to neutralize the virus. Okay, so basically what they're doing, they're transferring serum from one patient to another. Sometimes it works, sometimes it works a little bit less. But first of all, the IgM are, of course, pentameric, but are less specific compared to IgG. So I think that what we have to fix is a time frame and define what is the antibody that we need, okay, for that to cure the patient. What is the isotype, okay? Because then in the IgG, you can IgG1, IgG2, IgG3, and IgG4. And then, you know, it's a lot of work. And also as doctors, we always say antibodies, but antibodies, it's a word. Okay. Then as for the tests, so we have now this rapid tests that have been developed that are actually measuring IgG and IgM. So one of the main problems is that we were discussing before with Mushraf about that, is that you might have a cross-reactivity, for example, between the different coronavirus strain. Okay, so as for the spike protein, so basically one test can be positive, but because someone had a flu before and then just test positive for this. But I think that for now, so for the situation where we are in, and to get out of the lockdown, that is not a nice situation. I think it's useful because you can, for example, this is my personal opinion, you can for a second forget about the cross-reactivity and say, okay, this patient is symptomatic. We are in a particular time frame. So if it turns out positive now, probably it's COVID-19. Okay, then for the serology on a large scale, we need to work a little bit better, okay, in order to trace back who has been exposed, who has not been exposed, and the cross-reactivity at the time have to be resolved. I don't know if that answers your question, Mark.
Can I supplement on your answer, if you allow me, Marcello?
Sure.
So just to point out some of the 2 points, one very important point Ayat mentioned earlier is the samples in the stool. Now, we do have a paper from Anu Havrel, published this year, obviously, about the serology of COVID-19 and SARS-2 infections. And it appears in the stool, I said from day 6, but it appears even earlier from day 4, where you see stool swabs showing that you have the RNA of the virus in the stool. The other point I wanted to also implement some of the points you were mentioning about the antibodies. So it appears in this very elegant review, I suggest everybody to go and take a look at it in the Emerging Microbes and Infection by Lin et al. Recovering patients have neutralizing antibodies against the S protein on the SARS-2. And even patients with mild symptoms develop the IgG higher than those in severe cases, which is very interesting, I think, worth definitely looking into. Let's see, one of my students, Asil. Asil, can you hear me?
Yes, Doctor, can you hear me?
Yes, we can hear you. What's your question?
Okay, for people who die, if there's a possibility that the virus will be transferred from the dead bodies or not?
Oh my God, of course. Yeah, no, this is,
thank you, Asil.
Thank you really so much for this question. Yes, and this has actually been a problem in one of the cities that is close by to Bari, where I am. So in our region, there's 2 main cities. So one is Bari and the other one is up the north. It's called Foggia. So, and if you see the differential counts of the viruses, they have an outbreak more than we have. And one of the starting point was that there was someone that died, okay, died for different causes, okay? So in the elderly, you don't die for the coronavirus, but you die with the coronavirus, okay? But so basically they performed an autopsy, and then it turned out afterwards that it was positive. So basically there was a bunch of people that was contaminated with that. So of course, if I tell you this thing in this way, it might seem absurd, but I think that when it comes to these kind of challenges in medicine, we have to also take our time to basically, how can I say that, review all our procedures, you know, how do we work? And because we're dealing with something that we never had before. Okay, so before we didn't have to think about this problem, now we have to. And this point that you raised is absolutely right.
Ayat, are you still there? Hello?
Yes, yeah.
Yes, I am.
Can I ask you one of the questions, the very interesting questions that came from Prof. Adil and Dr. Tarf Al-Turki as well? She mentioned the idea of the hygiene hypothesis, that the people who live in a dirty environment or in the poor population, they develop, they have like a stronger immune response, while the people who live in more clean areas, they develop more allergic disorders. Do you think that can explain why the disease is not that spread in India or in Bangladesh, for example?
Now, that's one question we wanted to really jog your mind in, Marcello. We want to give you a proper exit.
Yeah.
Maybe just to give you more details about the idea of the hygiene hypothesis, that the people who get an infection with helminth parasites, for example, or with some microorganism, they induce Th1 immune response. While I think, correct me please if it's wrong, while in allergy they produce Th2 immune response. So it's exactly the opposite. And that's why we know from previous published papers that's why they can use the helminth, for example, antigens to treat the allergic disorder more than the other Th1 or Th2.
So it's so the hygiene hypothesis has been with me since the beginning of time, because it's you know, I'm dealing with allergy, so it is indeed as you said. So basically we have a branch of our immune response that we don't really use, that is the IgE response, that is the Th2 mediated response that we don't use for worms. So one of the hypotheses is that in our countries, so in the cleaner countries, if you want to say that, so we have more development of allergies because this kind of response is not used anymore. So,
so does that have an effect now with COVID, do you think?
So I think it's there are two different orders of questions. So the first question that we have to ask, for example, in the patients that have low level of lymphocytes, okay, is that, so the immune system is asked, so what do I respond or not? Okay, not responding is driving an infection really badly. Okay, so the point that Ayat is raising is the second kind of question. So once that I have a pathogen, how do I respond? Okay, so what you can imagine that, of course, you might have an immune deviation. And then I can tell you this. So, for example, even if you don't look at it in a large scale, but you look at it in a smaller picture, I've told you that Italy, it's shared into different portions. So the north of Italy is an industrial engine. Okay, so basically also the pollutants there are more than in the south of Italy. So we can indeed imagine that there's a problem in what we breathe, what we eat, the way we live influences the extent of an immune response. And with this, I'm totally right with you. But there's also another point, is that this is a disease that is affecting the respiratory tract. And of course, having, for example, a polluted environment and things like that might drive also chronic state of inflammation of the tissue that might give susceptibility towards certain kind of infections. And I can tell you that, and it might seem absurd to tell you that, but for example, in our unit in the hospital, we have on our roof of our department something, a collector for the pollens, okay, for the aerobiology. And I can tell you that since the lockdown, okay, since we have been locked down in Italy, for example, on these strips where we collect the pollens, you can really clearly see also the particulate, the smog, the pollution on that. And once that we have been in lockdown, everything is clear. So the amount of things that are actually affecting our lungs and our immune system in turn, it's really a lot, and you're completely right. I think that this might affect also the balance between the Th1 and Th2 response. But I think compared to allergy, it's a slightly different problem.
So what about the changes in the lifestyle nowadays that were everything, like we changed basically the lifestyle, so everything became more clear? Do we just prevent ourselves from the COVID-19 and put ourselves into a more risk of allergies, or it's not, doesn't work like that?
Well, I don't think it's so. I don't think it's a matter of one individual. Okay, so there's a genetic pressure, as you might know, on the immune system that makes that we develop in a certain way, and the HLA and MHC molecules are codominantly expressed from mother to father. So it's not, I think that is not something that we are dealing with as individuals, but it's for our offsprings, okay, for our children, eventually, that we can work on.
Okay, so we have a question from Dr. Tarfa. Would you like to take it, Mushraf?
Ayat, can I ask you for one more question from the crowd? Because I think Dr. Tarfa can wait. She's a good friend of ours.
Okay. Okay, what else? Some people also mentioned that, for example, Abrar, that sometimes the patient leave the hospital when not fully recovered, and they can still transmit the disease. What do you think about that? Is it because the low sensitivity, for example, of the detection of the viral load, and maybe that's why they are discharged, or they are really negative, but they can still transmit? I'm not really sure about the question. So what do you think?
Can I implement that question as well, Marcello?
Yeah.
People were talking about secondary infection of the same virus.
Yeah.
Have we seen that happening?
So I've read about it. I've never seen it happening, but I would lie. I've read it. So to answer your first question, I can tell you the protocol that we have now for example, I told you that there's a huge unit that is just dedicated to this COVID thing. Okay, once that the patients are negative for that, they will be transferred to another unit. And once that a couple of swab will be negative in, let's say, 4 days, they will be discharged. So there are no positive patient discharged from the hospital. So as for the reinfection, I've never seen it myself. In our region, we've never seen it. And if you want my personal opinion, I would find it unlikely that we fail to mount a proper immune response that protects us from reinfection. But we can consider mutation of the virus, and we can also consider that probably there are different strains of the viruses that are actually circulating with the same genome, because the diagnosis basically is based on expansion of a part of the genome. Okay, so probably you can imagine that there are two slightly different strains of the pathogen, and for some reason the antibodies that you're developing are not giving a full response towards the other. But this is my personal opinion, really. I don't take it for granted at all.
We have another colleague, Dr. Turki, who would like
Hi,
to...
Marcello. Thank you for an interesting talk.
Thank you.
So I have three quick questions, sorry.
Make them quick, Dr. Please.
The first one is, do DCs have the same ACE receptors as macrophages? As macrophages are was the main focus of SARS-CoV-1, the first SARS, and how the virus invade macrophages. Do DCs get invaded by the same receptor? The second question is, can you give me your explanation of the neutrophilia seen in immunocompromised patients with COVID-19? And finally, the fatal group of SARS-CoV-1 had the tendency of having a Th2 serum profile. Do we see this here in SARS-CoV-2? Thank you.
Thank you.
Thank you very much, Darfa. Thank you.
Thank you for your question. So, as far as the literature is developed so far, so the entry side is it's as we were telling before, is always the ACE2 receptor, and there are, to my knowledge, no papers that analyze the differential involvement, the differential infection, if you want, on DC and on macrophages. So on this point, we face the thing that we're dealing with an emerging disease and with an emerging topic in research. So as for the neutrophilia, that is a quite interesting point that you raised, especially in immunocompromised patients. And this is because so neutrophils are really I worked myself during all the years of my PhD when I was in France with neutrophils. So these cells are really peculiar. So first of all, whenever you have a tissue damage, neutrophils will get in. Okay, so this disease is a disease that is causing a massive tissue damage at the level of the lungs, and this will of course cause a massive influx of neutrophils. So secondly, as we were telling before, my guess about the neutrophilia is the fact that we were telling before that you have, for example, a raise in the D-dimer part, and this is particularly relevant, and we have to look at it because so D-dimers are connected, for example, to the complement system, and one of the main recruiters, I would rather say, of the neutrophils is the C5, that is a product of the complement cascade. And so we can easily imagine that during this infection you have a massive C5 production and thus influx of neutrophils and neutrophilia. Okay, so and what is interesting is that neutrophils at the end of the day are short-living cells, so they last, I think, 48 hours, not more than that. So it's something that is really sustained. So there should be something, either cytokine or something, that sustain the neutrophilia in these patients. So as for the Th2 profiling, I don't know if I picked the question correctly, but if you're asking me if we have done an immune profiling on the COVID patients in our unit, in our hospital, this has not yet been done. We're starting a project about that. And one point that I would like to raise with you as scientists and as immunologists is that we are only looking at how we are activating the immune system. Okay, so either Th2 response or Th1 response. One of the main point that has been ignored so far is the resolution of inflammation. So, for example, you might have heard that there was a moment also in the press where people were saying people that are undergoing treatment with non-steroidal anti-inflammatories are doing worse for some reason. So this is not something that we should ignore, because as you might know, so the arachidonic acid might go into different directions. So either in the 5-LO pathway that make leukotrienes and lipoxins, or in the cyclooxygenase pathway, okay? And this will go into the prostaglandin, for example, A2 and etc. So, but one of the main points here is that the same enzyme that is producing this, that are leukotrienes, is also producing something that is called resolvins or maresins, okay? And these molecules are actually shutting down the inflammation. So I think that a lot of literature, of course, but because as immunologists, we work in this way, so we try to look at how the inflammation is mounted, but we should look also on how it's actually resolved.
Yes, well, I mean, just to follow on the point with Dr. Tarfa's point, we do see an increase in different phenotypes of different cytokines, of different immune milieux, so Th1s, Th2, we see IL-10s and TGF-beta even though late in the infection. So it will be very interesting to see how this affects the timing. And I think this is the crucial question at the moment, the timing of the intervention and when to stop what cytokine at what time, because you have a 20-day, 2-week period where we need to direct our efforts to stopping the pathologies generating from the infection. I have a few more questions, hands raised, so I do ask you kindly to be patient with us. We have a lot of people who want to participate, which only suggests the interest.
I hope that is useful. That's just
very, very useful. I would ask everybody to be patient with us. I do understand the eagerness. It's all for the good. We have a very esteemed colleague of ours, Dr. Alia Dahlawi. Can you hear us, Doctor?
Hi, yes, Dr. Mashraf.
Hi, what's your question?
My question is that, first of all, thank you, Dr. Marcello, for your interesting lecture. I think you just mentioned now about this. We, as an immunologist, we are keen to understand the mechanism around all these immune cells and their mediators. I wish I have information if there is anything published or under study regarding the modulation of dendritic cells function by the virus, anyhow how these dendritic cells can be inactivated and unable to present their antigen to T cells. I just want to know if there is any information about this. I think you just said that this has not been investigated. I think it's very important to understand the mechanism of the infection. That's my question. And the previous one is about the cytokine storm. I think you mentioned that. Thank you very much.
Thank you, Dr. Ali. Thank you very much.
Yeah, so it's I turn to repeat. It's massive influx of literature now that everything is available online, and so as far as it is my knowledge, studies on maturation of dendritic cells has not yet been provided, but I agree with you on this point that is important, because so first of all, dendritic cells, if you're someone who's working with dendritic cells, have been ignored for over I don't know how many years, and that are the main core of an adaptive immune response is mounted. And this brings us also, would be interesting, because the question of one of the students was really interesting. So the infection either comes from the respiratory tracts or from the intestine. So what is interesting to know is that, for example, when it comes to immunization, if you give to mouse something intranasally or subcutaneously, you don't get the same immune response. Okay, so this suggests that the different subsets of dendritic cells tells to the immune system to make a different immune response. So I think that first of all, we have to understand on how the presentation to the adaptive immune system works, and that's correct. And then secondly, which of the subpopulation of dendritic cells are doing the job, and what the cells are actually telling to the immune system.
And the effective load as well, Marcello. I mean,
an effective load, of course. Yeah.
It's interesting that you've mentioned this. It was one of my recent lectures with the students where we talk about how the load of the infection, the number of viral particles can actually shape your immune response, whether to go pro or dampen the immune response towards it. Ayat, can you hear us? I know she's intently hearing us.
Yeah, hi. Yeah,
hi,
I'm with you, don't worry.
Any comments from the crowd?
Would you like to have a question from the crowd or from the chat?
Let's have it from the chat. What have people been writing and asking?
Okay. Well, there are so many questions about the asymptomatic patients. Do you think that they might be exposed to other viruses from the coronavirus family? And maybe that's why they are asymptomatic, or maybe that's why, maybe if it was reported before or not, I'm not sure, that maybe some people are developing resistance.
Yeah, I mean, it could be a possibility. At the end of the day, you know, if you want to make it plain and simple, our immune system recognizes shapes. Okay, so if for some reason there's a cross-reactivity in the shape of the of the spike protein of another coronavirus with the spike protein of this one, of course this will result as a milder infection. But this is the case, for example, also with influenza, you know, H1N1. When you make a vaccination, you will have people, even although the vaccination, some people will develop nothing, some people will develop a milder form of influenza. Okay, so I think yes. So if the question is, could have they been exposed to another coronavirus? Yes. But then again, it's we have also, I think, to look at it in a broader way. So we are all not the same. Okay, there's a huge polymorphisms among human beings. For example, just of the entry receptor. Okay, so the entry receptor is polymorphic. AC2 is polymorphic. The toll-like receptors that sense the viruses and the single-strand RNA are polymorphic. So it's you know, it's a complex cascade and then we have to gather everything together and say, okay, this individual is resistant for this kind of reasons.
I've got a question from a colleague, Dr. Hanan.
Thank you, Dr. Marcello, for very interesting topics. You have wrapped up the immunological point of view regarding COVID in very interesting and short way. I have a question. So now we have two approaches for COVID to deal with COVID-19. So the first is by developing a vaccine, and the second by focusing more in treating the syndrome, the cytokine syndrome, or the clinical syndrome from the COVID-19. So which one do you think is more critical and approachable and give a fast effect within the critical crisis?
Well, I think that so within a moment of crisis, it's of course the symptomatic treatment, the ones that has to be improved. Okay, so but in general, to develop proper treatments, and this is a broad problem with medicine. If I may, I can make an example. So, and it might seem distant from what we are talking about, but it's really, at the end of the day, you will see that it's related. So I mentioned before chronic urticaria patient. okay? So we had before in clinic, people suffering from chronic urticaria, they will have antihistamine, cyclosporin, second generation antihistamine, and all this cocktail of things. And we were, for example, asking a lot of questions about these patients. For example, do they have HIV? Do they have HCV? Do they have bacterial infections? Okay, and we were really running around in circles with this patient that were keep coming to our clinic with this chronic urticaria that was not going away. So at some stage, there was a guy that is called Maurer that discovered that actually these patients are making autoantibody that are of the class of that are IgE. So basically, these IgE are autoantibodies. So basically, if you treat this patient with something that is called omalizumab, the patient gets better, okay? So what I want to say here is that until the moment that this guy, so until the moment that Maurer found out how the system was working, so that actually it was autoantibodies anti-IgE, we were really in a hazy moment, okay? So these patients were really a mess for us. And this is actually the same for the COVID-19, in the sense that what is missing here, it's basic science, okay? So really understanding how the replicase is working, understanding which are the main targets, okay? As an immunologist, I can say, okay, the main target for a vaccine can be the spike proteins, but the biology itself of how this infection works is hazy. Okay, so I think that to make proper treatments, okay, symptomatic treatments, you can make them. But for example, antiviral treatment, to be developed, we need basic science, okay? So this is really something that you're just now. And then in the development of the vaccine, I think that is something that we can deal with in a second time. But we can take advantage of this, quote unquote, take advantage, of course, because it's a bad situation. But during pandemic, as far as I know, the development of drugs and of vaccines do not have to go in phase 1, phase 2, phase 3. Okay, so once that you have something that more or less works, you can directly try it on human beings. And this is really something that is peculiar for pandemic states. Normally, it's not allowed. I don't know if I answered your question.
We have a question from a colleague, Dr. Fadi Kashgari.
Yep.
Yes, Dr. Fadi, can you hear us?
Hello. Yeah, hello.
Hello.
Yeah, sorry. Hi. Yeah, regarding to Ayat's question earlier about the comparing between the developed country or rich country and poor country, there is a paper being published recently, I think, has noted about the BCG vaccine, which usually being given in poor countries like Pakistan and India and African country, while it's been stopped to give in a rich country like Italy. Obviously, they said there is a correlation between the BCG vaccine and the low fertility rate of COVID-19. What do you think from your point, from your view, I mean, as a clinical? Do you think this is could be happen?
So it's of course we can correlate the things with the COVID outbreak with different things. So the BCG vaccine, it's a particular vaccine that has been always surrounded by sort of particular thing, because it's not effective 100%, okay? It's not, so the BCG vaccine, as you might know, it's not made up of the strains of the BCG, but it's an alternative mycobacteria that is supposed to give, let's say, a cross protection. And it's actually, if I'm not wrong, it's effective in 65 or 70% of the patients. So if one may ask, okay, so once that I had the BCG vaccine, I had, imagine, more type 1 response ongoing, so I have more interferon that I might produce, Okay, so at this point, I would say, okay, if this correlation works, why not? Okay, we can make BCG vaccine on a large scale, but we can also bypass this and say, okay, so what about the role of interferon in all of that? I know that this might sounds absurd, but you know, cytokine are quite powerful tools that we have, and I think that we don't use it that much.
Ayat, are you still with us?
Yes. Yep. Do you think we shall take Dr. Ahdab's question?
Yes, of course. Another immunologist.
Yes.
Are you okay with time, Marcello?
Yeah, I mean, I'm home, it's fine.
Okay. Yes.
Yes, hello, hi. Thank you very much for this webinar. We've enjoyed it very much. Time is very important because I do have a couple of questions as well. So the first question about the T-cell exhaustion, because it has been shown that T-cell exhaustion is exists, especially in severe cases of COVID-19, in COVID-19 patients. So do you think releasing the T-cell break in combination with anti-inflammatory therapy would help?
Mm-hmm.
This is the first question. The second question is that you said, because you draw a line, you said the infection came from bat to human, but it can go back. So do you mean that the infection from human to animals is not possible? Just to confirm before.
So I'll start from the second one, that it's easier. Probably I was not clear on that. So it's and I'm sorry for that. So it's just that the phenotype of the virus as such that has developed the ability to infect human cannot infect back. But of course, you can have then by another intermediate. Of course, you can flip back, but it will be another phenotype of virus. Okay, so the point is that the infection is restricted to either humans or the intermediate or the bats.
Like many infections, it's species restricted.
Yeah.
We want to know your thoughts because there is a Chinese study in cats, domestic cats, has been tested positive for human COVID-19, and also as well the Malayan tiger in US that has been believed to be infected from asymptomatic zookeeper carrying the virus.
I would like to know how they took the sample, the nasopharyngeal sample from the tiger.
This is my third question, by the way.
Can you tell us, Marcello, how you can take it from tigers?
Well, so concerning the fact that COVID-19 can go back from humans to cats, it's indeed something that is sorting out now. The data that I have it's coming from 2013 on Nature about COVID SARS-CoV-1. Okay, so the fact that it cannot infect back the bats. Of course, at the time it was not such a huge pandemic, so I don't think anyone had looked at the time whether it can infect domestic animals or things like that. So I think it's a major challenge, and it's important to develop that. So concerning the T-cell exhaustion, it's an interesting thing. So what you propose is that to give anti-inflammatory, and I think I missed the word.
The checkpoint blockade, like to release the T-cell break, because it has been shown that PD-1 expression and TIM3, which is the exhaustion markers for T-cells, is upregulated, especially in severe patients.
Yeah. So what can I tell you is that the idea that I have, and then there has been some recent papers also about, so the anti-PD-1 things has been extensively used, for example, in the treatment of hepatocellular carcinoma. And so I think that the phenotype of exhaustion is not widespread. So, for example, if you take some of the recent paper on HCC, and I know that because my wife was working on that. So basically, within the tumors, the T cells are exhausted, but the phenotype is quite different on the bloodstream in the periphery. So it's what we have to figure out is where the cells are exhausted. Okay, because you can imagine also different ways of delivering the drugs, of course. And it has to be confirmed that peripheral lymphocytes are actually exhausted, and these are the ones that are going to do the job in the periphery. I don't know if you get my point.
Yeah, I got the point.
The point is quite clear. I'd like to add one more point. It's proving now challenging, especially with recent papers now, to identify lymphocytopenia. Because are you missing, because they're looking at plasma, they're looking at the blood. Are they missing the recruitment of these lymphocytes in the tissue? It's always a challenge because, as you know, Marcello, in research, when we want to identify a drop in numbers or a lack in counts, we associate tissue staining with either lymphocyte counts in the lymphoid or blood lymphocytes. So to jump to a lymphocytopenia conclusion in future publications, I would hope, because I'd love to see how these neutrophils look in the tissue when we don't find them in the plasma. And I'd love to see how the lymphocytes look like in the tissue, because they are recruited. They are going somewhere. They can't drop and disappear.
Yeah, sure.
Would that be because of the virus using another receptor rather than ACE2?
Yeah, but I mean, it's for example, I was telling before that as for the COV1, for example, another receptor can be used, that is the PP4. That is easily expressed on lymphocytes. So it's the anthracite might also trigger problems in immune system.
We're seeing about CD147 as well on T cells.
Yeah, exactly.
Well, I mean, when incubated with the virus, cells overexpress ACE2. So the virus has no problem finding its way into cells. That's where vaccines come into question, because we've had so many candidates for vaccine: the S protein, the proteases, the protease molecules, and the ACE receptor itself. So that leads to many questions. Let's go to Ayat. Ayat, are you still there? Can you hear us?
Can I ask my last question? Am I allowed to?
You are allowed to, Adab.
Okay, thank you very much. So I came across a question yesterday on Twitter, and I think it's a very interesting question about if anyone has compared saliva versus the nasopharyngeal sample that we are taking now for COVID-19, because it will be easier to detect the virus, I mean. So
Yeah,
how does this in Italy? I don't know. Or...
So as far as I know, and this is also in the guidelines for the clinic, so it's required, for example, to make both, for example, the swabs, also the oropharyngeal and the oral swab, because as you say, so the nasopharyngeal that goes from here to inside is more precise. Okay, so probably this deals with, I think that this deals with how the virus enters our body and where it can, stick and then infect the cells. And another important point about the nasopharyngeal thing is that you might have heard about it, but there are a lot of patients of symptoms that they refer to us is that they don't smell anymore, so they can anosmia. Okay, so probably there's something to look at in this entry point. Okay.
Are you thinking of desensitization, maybe something to do with the nerves, nerve damage?
So there has been a paper, but I was not convinced about it. Is that because it was in mice? So basically, there were, and I think I can send you that afterwards. I think so. Anosmia might be caused to different things. So basically, either you have a chronic inflammation in the nose, and this happens also, for example, with patients affected by rhinitis, or that the virus somehow is allowed me this word, walking on the on the nervous system. So in mice, it has been demonstrated that somehow this COVID-19 has the capability to have this neurotropic effect, but so far it's not been demonstrated in human.
So just to follow on the point you mentioned about remdesivir, I know you mentioned that your colleagues have not gone on with it. There's a new paper in the New England Journal of Medicine, Jonathan Guerin, an MD, and they show that nearly 68% improvement in very, very, very sick patients. So very sick.
With what? With I missed the word.
Resdemivir, which is an antiviral.
Right. Yeah. I know that 60% it might sound a lot, but I think that in our unit has been shut down because there was this contradiction in the literature. Okay, and then I personally, I don't know what is your opinion about that. What is your opinion, guys, about that? Is so that is that these two antivirals are specific inhibitors of replication of HIV. So we are using these molecules. I have hard time understanding why. Okay, so at this point, I can tell you why at this point not using another antiviral that we have. You know, it's, you know, I have a little bit hard time with the story of the antiviral. I don't know, what is your opinion on that?
Well, from my understanding with the antivirals, it mostly prevents the RNA polymerase functionality. They all address the intracellular cascade of events happening inside the cell. Now, I'm not a virologist, so I don't know how important that is in the viral replication of viral involvement. I understand it's hard already, and we have very strong elements in the immune system that deal with intracellular infections and cytokines that deal with intracellular infections. But how these monoclonal antibodies detect the protease activation, how they prevent RNA polymerase activation, I don't know. That's possibly for somebody who's more into the pharmacokinetics of the disease. Ayat, can you hear us?
I guess the last interesting question that we can end from the audience with came from Dr. Salma. She asked if we can manipulate the macrophages by, for example, inhibiting M1 macrophages or inducing M2 type macrophages can help in case of the chronic inflammation?
Now, that's a question for Ayat specifically. I know how much you like macrophages, Ayat.
Well, I'm not a macrophage person. I'm more into a vaccine person, but I think it's a very interesting question.
And you do come from the school of Catherine Else, who's a genius in macrophages.
Yeah, that's true. But I really focus on DCs more than macrophages on my project. But so I won't call myself a macrophage person.
Marcello.
So it's that is very interesting. It's so of course you can manipulate macrophage phenotype. So the M1 or M2 phenotype. So in one of the way that in an experimental setting this has been done, it's via using the capability of macrophages to pick up to phagocytes. So these are phagocyte cells, okay, and you use the capability of macrophages of picking up things, basically. And the technology that has been developed, for example, is the technology of nanoparticles, okay, that is not dealing with phagocytosis, but rather, they just get in the things. But since you're dealing with nanoparticles, okay, this is working in vitro. Okay, you can manipulate the phenotype of a macrophage from M2 to M1, for example, from M1 to M2, and your readout that you use is basically what panel of cytokines are these macrophages producing. But the main point with it is that these main mechanisms, okay, that I know, so for example with nanoparticles or with encapsulated things, is that this particle will be picked up by any cells of the body. So there's again a problem with a broad effect on all the body. So this kind of, for example, studies have been done with the purpose of manipulating the macrophages that are invading a tumor, and because of course you want a less tolerogenic phenotype in that, but the problem is the drug delivery in that case. So it's to specifically target macrophages. And as far as I know, there are no tools to specifically target the macrophages and make them have a drug that you want inside.
Like dendritic cells, I think we haven't brushed much on macrophages, which I'll make sure in future webinars that we can talk about.
It's beautiful, by the way.
I mean, of course, I agree with you. The idea of the involvement of macrophages, I mean, Zhu et al. In prospective immunology suggested that the source of IL-6 and interferon gamma driving Th1 immune response are granulocyte microphage colony-stimulating factors, so GM-CSFs. And he sees that specifically monocytes, CD14, CD16, CD45, drive the GM-CSF factor towards the Th1, Th2 in COVID-19. So that's possibly an interesting note for future mentioning. Let's take one more question from Faisal, a colleague of ours, if you don't mind. Faisal.
Hi, Marcello. First of all, thank you for your informative presentation, and thank you, Mushrif, for hosting this webinar. I have a couple of questions. Sorry for maybe the time is not good. The first one is we know that the virus can interfere with the interferon production, but I was wondering if there is a publication shows that phenotype with the SARS-CoV-2.
Very good question. Very good question.
And as you have a clinical background, what is the best time to administer the anti-IL-10 receptor? Maybe,
sorry.
I didn't catch it earlier. Thank you.
What's the better time to administer anti-IL-10s? Because you see IL-10 increasing.
As far as I know, no. There's no timing. But I turn to repeat all the protocols. We're dealing. So there's this huge fracture in general in science and medicine, so that the translation to practical medicine is quite slow. So the protocols as for this COVID-19 are still developing, if you want. Okay, and then what you have to know that, as for example, for the sanitary system as it's conceived, for example, I can speak for our countries. There's a lot of variability, for example, between region and region. Of course, we more or less have guidelines, okay, but inside the guidelines we can make different things, okay, so according to the clinical state of the patient. So, and concerning the first question, if I got it well, is that viruses might alter the interferon production upon the first sensing cell. So it's in my knowledge, this has not been demonstrated for the COVID-19, and it could be a possibility, but we have to remind. I know that it might sound repetitive, but so the single-stranded RNA goes straight directly, is sensed by the toll-like receptor 8. That is something that is quite potent stimulator of the immune system. So it's then. I would rather think that it's a secondary mechanisms upon viral replication that eventually dampens the interferon production. But the first kick, of course, interferon is produced.
So just to implement the point that Faisal brought up, so Huang et al. Clinical features of patients infected with the 2019 novel coronavirus in Wuhan, China. They've shown, they've looked at many cytokines, IL-1 beta, IL-1 alpha, IL-7, yeah. many chemokines and many coagulation factors. And one of the cytokines, IL-10, they showed an increase in ICU patients. Mm-hmm. Marcello, this has been really enjoyable. I can't say how much we're glad to have you here. You've been more than generous with your time. Do you have anything at the end you'd like to say?
No, really, guys, thank you so much. I will leave you my email address here for the ones that might be shy. Okay. That is this one, marcello.albanesi@uniba.it. And really thank you, Anayat, for hosting me, for having me. And I think that in general, we have to take what is good in this pandemic, in the sense that we now are forced to use this kind of platform to share knowledges. And I think that we should keep doing it over time, you know, and this can be really a bridge, and especially for the students, for the people who are learning things, you know, it's, I think it's important. We should continue doing that.
This has been one of the longest webinars we've had, close to 3 hours, and that's all for your generous time with us, Marcello.
No worries.
Next week, guys, we've got Dr. Joanne Pennock. Arrivederla, ciao ciao.
arrivederci.
مع السلامة.
عفوا.